Next speaker today is Dr. Bruce Korf. Dr. Korf is the Wayne H. and Sarah Cruz Finley Chair in Medical Genetics and Professor and Chair of the Department of Genetics, Director of the Heflin Center for Genomic Sciences at the University of Alabama at Birmingham. Dr. Korf?
Thank you very much. Pleasure to have an opportunity to participate. I've been asked to address the interpretation of genome sequencing data. First of all, these are my conflicts to disclose. And I'm going to begin with a purely hypothetical case study, which I hope will set the stage for what I'll be talking about. So this is a young child who's got developmental impairment and some mild dysmorphic features, recently diagnosed as having autism spectrum disorder. He has a cytogenomic microarray done as part of his evaluation, and this is normal, but then it's suggested that exome sequencing be done in order to help provide an etiological diagnosis. A not particularly uncommon scenario, as genomic testing is being extended into other areas, including pre-implantation diagnosis and the non-invasive screening. Some of the same kinds of issues, as we'll talk about in the coming slides, will arise. So if you think about the workflow of genome sequencing as it's applied clinically, you can very broadly divide it into three major components. That of pre-test counseling, talking to the patient or the family about what the testing can and can't do, what the potential risks and benefits are, is critical. This is not the kind of test that you draw a sample on somebody from, send it to the lab, and deal with the nuances of interpretation when results come back. The patient really needs to be prepared with clarity as to what they can expect and to be ready for the possibility that findings may occur that would not have been anticipated. The second stage, of course, is the actual testing itself and the data analysis. The testing platforms are evolving extremely rapidly, and the time and effort required is truly a moving target. But once testing is done, then there is a need for data analysis. And this is increasingly proving to be the most complex component of the process, because hundreds of thousands, millions maybe, of genetic variants will be identified in anybody, regardless of the indication for sequencing, and sorting through those to figure out which ones are important and relevant clinically and which ones are not is by far the most challenging component of the process. And then once all of that is done, there is the need to interpret the data and to report it clinically. The interpretation is done in part in the laboratory, trying to filter the variants, but also in part by the clinician, who is the one that is in the best position to identify things that are directly relevant to the care of the patient. And of course, it comes down to the clinician who's most familiar with the patient's clinical situation to actually report this back to the patient. So let me dig down a little bit into each element of the process and begin with the pre-test counseling. I guess the first question is, why would you want to do genomic sequencing? And in at least a context of an individual who is already born, usually it's either the presence of an unknown genetic diagnosis, somebody who has signs or symptoms of disease, but in spite of efforts with conventional approaches, a diagnosis hasn't been established, or it may be done in the setting of an individual where there is a clinical diagnosis, but where the differential diagnosis from a genetic perspective is extremely broad, making it impractical to do testing on a one gene at a time basis. So the exome sequencing offers the possibility of testing across the whole genome quickly and gives you much broader coverage at much lower cost and with greater speed. So the benefits of this, and by the way, it's worth mentioning that if this is applied in either a PGD context or a non-invasive prenatal screening context, you obviously don't have an a priori question about what particular condition you might identify unless there's been some kind of evidence either from the family history or from perhaps an ultrasound to suggest a particular condition of concern. But otherwise, on a screening context, this is being done without any particular a priori question. Anyway, the benefits from a symptomatic individual's point of view is that finally a diagnosis may be established, and it can be done at lower relative cost. And I mean by that, the actual test itself may not be less expensive than any one particular genetic test, but in the aggregate, when you consider the number of individual tests that you might have to do, this is answering in one test what otherwise might have taken multiple tests to do. And of course, there's the advantage of rapidity because you're not sending a given test, waiting weeks to months for it to come back, only then to need to send yet another test. And to do this in a kind of serial basis can actually take months to years, whereas when one does the genome, one is essentially looking across all the genome at one time, so it can significantly increase the speed. It may not be faster than testing just one gene, but it's certainly faster than testing one gene after another for a long period. Now, on the other hand, there are risks that the patient needs to be alert to. The obvious one is cost. Again, it may not cost as much as doing 10 tests, but it does probably cost more than doing one test, and making sure that the costs will be covered by insurance, for example, is an important point. And increasingly it is, although also it requires a lot of care to set it up with appropriate indications and to get pre-authorization. That's the obvious risk, but the other risks, at least some other risks that one needs to be alert to, first of all, the possibility of secondary or incidental findings. Those might be synonymous to some people, where secondary may mean looking for things that are not the primary diagnostic indication, but which are still thought to be important. Incidental could be defined as finding things you're completely not expecting, but just stumble upon. But either way, they boil down to the same basic thing, which is to find something that is medically significant that wasn't the primary indication for doing the testing, but which could have implications for the care of the patient. And I think the other main risk is that of finding a variant of unknown significance. I guess it's an obvious risk that you might not come up with an answer, because there's no guarantee that genome sequencing will find every possible genetic variant, or even that the condition at hand is genetically determined to begin with. So that's a sort of obvious risk, I think. But the less obvious risk that many patients aren't prepared for is that you may indeed find something, and it will be something real, but it will not be something that necessarily is clinically significant. And actually knowing whether it is or it isn't clinically significant even can be tricky in some cases. I'll talk about some of the approaches to doing that in a few minutes. But it's important in pretest counseling that patients realize that there are some sort of gray zone results, at least currently, that can't always be interpreted with confidence. So what we're talking about in general is the concept of massively parallel sequencing, and I think other speakers have covered that, so I'm not going to dwell on it. This is a diagram of one of many approaches, and in fact, the truth is that this is a moving target, and the exact platforms evolve. In essence, what you're doing is breaking the genome into very small parts, sequencing each one of them in parallel, and although these may be only 100 base sequences that you're obtaining, in this case by taking templates of DNA from the patient and using them as the template for a DNA synthesis reaction, and basically color-coding the bases and taking a photograph of which base is incorporated into which fragment at each time a base is added, and so piecing together the sequence from the photo. But whatever the platform exactly is, and the chemistries evolve and are different in many different ones, fundamentally what they have in common is this concept of massively parallel sequencing, looking across the whole genome in relatively short fragments, but simultaneously. So it permits you to be doing the DNA sequencing with an enormous increase of efficiency. Question could come up as to the difference between exome and genome sequencing. The exome, the component of the genome that actually encodes protein, is something between 1 and 1.5% of the whole genome, and so far appears to be where most of the clinically relevant mutations reside. So you can save time and money in terms of sequencing by focusing on the exons, which are, in other words, the exome, as compared to the genome. Probably as the technology continues to improve, it will become less and less necessary to do this focusing, and I suspect that the sequencing of the genome will eventually become more common. But for now, exome sequencing is clinically more often used. Well, what about the analysis? This is a screenshot from a paper that Heidi Rehm and I wrote for JAMA earlier in 2013. And it begins with the pipeline of isolating exomes from the DNA and then sequencing them, what we've just talked about, comparing the sequences that are found in these exons with standard reference genome sequence to figure out which are the child's variants, and then filtering these by comparing the variants in the child with databases of variants found in the general population. And the presumption here is that you're dealing with a relatively uncommon condition and one that is therefore not represented as a variant in common catalogs of common benign variants. So you filter out the common variants and are left with only those variants in the child that are considered to be not common or rare. Now, in order to do further analysis, you have to have some idea of the mode of inheritance, and there are depicted here a few possibilities. One is that it's a dominant being passed from generation to generation, and that there's an affected parent as well as an affected child. And if so, it's really helpful to test all three. And what you're looking for is a mutation that's present just in one copy of some gene and is represented in both the affected parent and in the affected child. Skip over to the far right. Another possibility is you think it's dominant, but you think that neither parent is affected and that the child represents a new mutation. And in this case, what you're looking for is a mutation that affects just one allele in the child, but is not represented in either parent, and hence is a new mutation. An autosomal recessive implies that the child have a mutation in both copies of a gene. Now, it's not likely to be the same exact mutation, but it's some pathological mutation that affects both copies of some particular gene, and each of the parents should be a carrier for one, but not both of those, and that would be the kind of scenario. So you do your filtering, pulling out the common variants, and then looking for these patterns that would suggest familial transmission. And if you don't know the mode of inheritance, you may have to experiment with all of these possibilities. Ultimately, then, you may end up with a short list of genes that fulfill the criteria, depending on what you think the mode of inheritance is, and you have to begin to analyze them to see if they make sense in terms of the child's symptoms based on whatever might be known about those genes, and also to take a look at the variants and see if they would be predicted to have an important effect on the gene or the function of the gene or the function of the protein. Many benign variants exist that have no impact on the function of a gene or the function of the protein it encodes, and those would be poor candidates for a disease gene, whereas if you can model this using a variety of computer modeling approaches, you can estimate the probability that it would have a disruptive effect on the function of the protein. You can look to see if the variant is at a site that's highly conserved in evolution, but one way or another to evaluate variants, ultimately hoping to identify just a specific gene that you believe is associated with the condition in that individual. And ultimately, vis-a-vis at least the original question, that's the result which is reported to the referring doctor. This is, of course, an intimidatingly complex slide, and I'm not expecting to walk you through all of this. I want to simply point out this is a decision tree published in Genetics and Medicine a few years ago to validate particular variants in terms of whether they are or are not likely to be associated with a particular phenotype. Sometimes it's fairly obvious that a variant either is or isn't phenotypically significant, but other times it can be not so obvious and requires a fair amount of digging. And over in the left-hand corner are some of the kinds of evidence bases that might be used to support the pathogenicity of a protein. This is summarized here. You can scan databases of known mutations in a particular gene to see if the mutation that has been found has been seen before. You can verify that that mutation is generally, if it's thought to be pathogenic, not seen in unaffected individuals in the population and seen in affected individuals, although the latter may not be easy if it's a rare disorder and a particular variant has just not been reported before. You look to see if that mutation's ever been seen in trans with a known pathogenic mutation, because if it has, that means found on the other chromosome with a known pathogenic mutation. And if you're dealing with a dominant at least, that would argue that the mutation you have found is probably not clinically significant because you found a different one that is clinically significant. And if you have other cases like that, sometimes it will be helpful to show that a particular variant is not significant. If you have the opportunity to determine if the mutation segregates with the disorder in a family, of course that requires an extended family, that can be very helpful. From a perspective of computer analysis, you can see if the variant is conserved at a site which is conserved through evolution. Certain amino acids can vary between different species and don't seem to change the function of the protein, whereas others may be highly conserved, and the latter, of course, are the ones that would be at greatest likelihood of being clinically significant. And finally, there are so-called in silico prediction programs that look at the structure of a particular protein and how it might be altered by a particular mutation and whether that indicates pathogenicity or not. Well, what about the possibility of incidental findings? I use this as an example of the concept of walking by and seeing a passerby have a flower pot fall on their head. And I think most of us would agree in situations like that that there's a certain responsibility to warn individuals if you can prevent harm by doing so. And I think that's the spirit in which I would approach the issue of incidental findings. Earlier in 2013, the American College of Medical Genetics and Genomics published a guideline, recommendations for reporting incidental findings in clinical exome and genome sequencing. Incidentally, it's not intended to cover research-based testing. And I'll walk you through some of the major recommendations as they apply in the clinic. They, first of all, identified a minimum list, 56 disorders. I'll show you the list in a moment, where constitutional, we're not talking about acquired cancer-related mutations, for example, but constitutional or germline mutations should be reported by the laboratory back to the referring doctor, regardless of patient age, and that furthermore, that labs should seek and report specific types of known pathogenic variants in genes that are on that list, with the ordering clinician responsible for pre- and post-test counseling in terms of the incidental findings. So this is the list, and I'm not going to walk you through all of the conditions on the list. They include a fair number of tumor predisposition syndromes, several connective tissue disorders, a variety of cardiomyopathies and arrhythmia-related disorders, familial hypercholesterolemia, and malignant hyperthermia. And the principle that led to the inclusion of any particular gene or gene variant was that, I think this is summarized on the next slide, or some of the questions that we raised. Why did we look at these particular genes? Have we given up on patient autonomy? Why should we test children for adult onset conditions, and why should we do this now? So back to the criteria for inclusion, there needed, first of all, to be a confirmatory test so that the result could be used as a basis for clinical decision making. There needed to be preventative measures and treatments available that would alter the outcome in a positive way if that variant could be identified early. There needed to be a significant asymptomatic period, and the importance of this was that things that already should have been diagnosed clinically are not included on the list, because the point of this is to find things you otherwise wouldn't suspect. But things that you should have figured out already because they would have been symptomatic are not on the list, on the assumption that you already would have known that these are there, or at least something related to these conditions affected the patient. These had to be mutations with a very high likelihood of causing disease, where we have good evidence of very high penetrance, so that we're not just telling you this is something maybe, possibly you'll someday get, but where the chances of actually getting the disease were very large. And finally, these had to be very well documented pathogenic mutations, not variants of unknown significance, certainly. And this was not intended as sort of a test bed to see if things were pathogenic. It was really intended for things that were already documented very clearly to be pathogenic. So you could imagine an example, and this is one that comes up often, of discovering a mutation for hereditary breast and ovarian cancer, which could occur at any point in a genome sequence, whether it's done prenatally, postnatally, or at any time. This is a condition associated with an autosomal dominant transmission of significant risk of breast and ovarian cancer, as well as a variety of other cancer types. Two major genes with appreciable frequency of pathogenic mutations, and well-documented examples of risk reduction. It's an example of a family. This is not an example from genomic screening where an individual was concerned about her risk because of a family history. A sibling was found to carry the mutation, and she was not, which tells you that she indeed is not at risk of developing cancer. So the spirit of these recommendations was that if you found this, you would be able to warn somebody that they were at significant risk. And many people come from small families where there might not be sufficient family history to have raised the concern before. But once identified, there's a very clear path to offering risk reduction strategies of a variety of different types. What about testing children? Because if this is discovered in a child, then you're in effect testing a child for an adult onset condition. And a joint statement from the American Academy of Pediatrics and the American College of Medical Genetics and Genomics recently addressed this question of genetic testing in children and actually reaffirmed a long-standing view, which is that the preeminent concern should always be the best interests of the child, both from a physical and psychological perspective, and also maintaining the privacy of the child and the appropriate social environment, for example, not stigmatizing a child with information about a condition of adult onset, to only test the child if there were clear medical benefits that would accrue to that child at that point in time. And if we're dealing with an adult onset disorder, to wait until the child is grown up to be able to offer informed consent. You might say, here's an example where maybe you would discover a BRCA mutation in a child. We ordinarily don't recommend testing before 18 years of age, but the argument here is to the justification is this is not a case where you're testing a child for a known familial pathogenic mutation. Generally, if it's an incidental finding, it's not expected, and it's a child in a family where they don't already know that there's risk of a BRCA mutation. And does the child benefit? Well, if the child is a carrier for such a mutation, you can expect one of the parents must be too. And if there's no known family history, who knows when that risk would be recognized? And the child, in fact, does in effect benefit right then and there, mainly by virtue of having a parent who remains healthy. And so this is really very different from the scenario of a known family history where it can be in the back of everybody's mind to test the child when he or she grows up. Here, that may never happen if you don't identify this incidental finding, because there's no guarantee that the family otherwise would know that they're at risk until somebody actually develops cancer. You should know that very recently, in 2013, the Presidential Commission for Bioethical Issues has issued a report. The screenshot of the cover is shown here, And fundamentally, I think they agree with the concept of the need to be alert to incidental findings. They make a strong statement about the need for pretest counseling. There is a consideration that patients be given an option not to receive test results. And there may be circumstances where, for example, an elderly person with metastatic cancer and no family members who could ever benefit might rationally not want to know about results that literally will have no impact on their future health or on the health of their family. So the point they make there is that it's important for the clinician who's doing the counseling to inform the patient of what they can expect and to respect patient choice vis-a-vis the possibility of incidental findings. So I think there's a lot of further discussion on this point that needs to take place. So you could ask the question, where are we going with this? I think we're going to see an increasing clinical application of genome sequencing, certainly in the instance of symptomatic individuals, increasingly also in the instance of identification of individuals early in life, and as we increasingly do both pre-implantation diagnosis and newborn or prenatal screening, these kinds of issues inevitably are going to come up. The incidental finding list is subject to continual revision. It's truly a moving target, and the American College of Medical Genetics and Genomics is instituting a process to accomplish that ongoing review. There will need to be outcome studies of the application of genome sequencing and also the recommendations for incidental findings. And there is also a funded now effort to build a clinically relevant variants resource, because there is, I think, a lot of work that may take a long period of time to be completed to fully annotate the genome. We are very much in the early days, and our ability to sequence genomes has far outstripped our ability to interpret all the results. Now, that is not to say that there is no clinical utility now. There indeed is clinical utility. As we speak, many diagnoses have been made that would otherwise have eluded diagnosis probably indefinitely. So this is here and now, and we have to respond to this opportunity in the here and now. But that said, there are 22 or so thousand genes, untold numbers of genome variants that need to be annotated, and this is going to take a long period of time with systematic effort to reach the point where you can look at a genome sequence and achieve the kind of predictive value that all of us ultimately would be hoping for. So going forward, I think we can expect to see increasing usage and increasing clinical utility as the databases improve and are further enriched. So in summary, pre- and post-test counseling are absolutely critical in the application of genome sequencing. The patient and the clinician must be alert to the possibility of incidental findings and also the possibility of revealing variants of unknown significance. Genome sequencing, especially exome sequencing, but genome sequencing in general, does not detect all possible mutations. Copy number changes, for example, are not detected by most approaches right now, but there are undoubtedly regions of the genome that will be difficult to analyze, so this is not a 100% ascertainment, but it should be recognized that this can provide increasingly a practical solution to diagnostic problems in a cost-effective manner, and without question is the wave of the future and indeed the wave of the present in terms of medical genomics. Thanks very much.
All right, thank you, Dr. Korf. I have a question. We mostly discuss single gene disorders during your talk, but I assume next-generation sequencing is equally able to detect whole chromosome gains or losses or large structural changes that are already identified on many other platforms for prenatal screening. Is that the case?
Well, the prenatal screening approaches have mainly been focused on trying to find usually extra chromosomes, especially trisomies 13, 18, and 21 associated with live-born significant clinical disorders or clinical syndromes. And there are other approaches to finding these kinds of changes, but if one wants to use the fetal DNA in the maternal circulation, the use of next-generation sequencing really has proved to be of key importance for doing that. So that's really the motivation, at least the initial motivation for using next-gen sequencing in this context. In principle, the same approach, however, could be brought down into a much finer level of analysis, looking at individual gene mutations. And that is not, at this point, the sort of focus of non-invasive prenatal screening, where you really do have the potential to, on the one hand, diagnose a greater variety of disorders. On the other hand, open Pandora's box in terms of all the various things that you might find, many of which may be even of unknown significance. So I guess we have to be alert to the fact that a technology that has been really focused on the most sort of high-level analysis has the potential, probably first in cases where you have reason to suspect a single gene disorder, but in the long run for much broader screening. And I think that's the complexity that we're going to need to be prepared to handle.
All right. Thank you very much. Appreciated your talk.